This content is for informational purposes only and does not replace professional medical advice. Always consult a qualified healthcare provider for personal health concerns. Have you ever wondered why a news headline claims a new medication is a breakthrough, while your doctor remains cautiously optimistic? It is a common experience to feel caught between the excitement of a medical discovery and the reality of your own health journey. Behind every headline, every clinical trial, and every treatment option offered in a sterile exam room lies a complex world of mathematics that dictates how we interpret success. We often assume that if a study shows a drug works, it is a definitive fact, but the truth is far more nuanced. Researchers rely on a specific statistical tool to measure the reliability of their findings, a tool that acts as a bridge between a small group of study participants and the millions of people who might eventually take that treatment. Without understanding this piece of the puzzle, it is easy to misinterpret the potential benefits or risks of a health intervention. We are going to peel back the curtain on the language of clinical research to reveal what those numbers actually tell us about the safety and efficacy of medical care. By the end of this journey, you will possess the clarity needed to read health reports with a discerning eye and a deeper understanding of how evidence is constructed. It is not just about the final percentage of success, it is about the range of possibility hidden within the data. Throughout this video, we will dismantle the mystery surrounding statistical significance and move toward a more grounded perspective on how evidence-based medicine truly operates. As we transition into the foundational concepts of this topic, we will begin by examining the specific mathematical framework that makes these conclusions possible, shifting our focus from the broad claims of public media to the precise mechanics of scientific analysis. This content is for informational purposes only and does not replace professional medical advice. Always consult a qualified healthcare provider for personal health concerns. To understand what these numbers actually tell us, we have to move past the idea of a single, perfect answer. When researchers report findings from a clinical trial, they aren't trying to pinpoint one exact value. They are trying to capture the truth within a bracket of probability. Think of it less as a measurement and more as a safety net. Imagine you are trying to estimate the average height of a crowd from a distance. You wouldn't guess one specific inch. You would identify a range, say, between 5'6" and 5'10", where you feel reasonably certain the actual average resides. A confidence interval provides a range of values that is likely to contain the true effect of a treatment. If a study suggests that a new medication reduces blood pressure by 10 points, the confidence interval might tell us that the effect could realistically fall anywhere between 6 and 14 points. That span represents the uncertainty inherent in sampling a small group of people to represent an entire population. The width of this interval is a direct indicator of precision. A narrow range suggests that the researchers have a high degree of certainty about the treatment's effect. Conversely, a wide interval signals that the data is less definitive, perhaps because the sample size was small or the results were highly variable. It is a way of quantifying our humility in the face of scientific complexity. By focusing on these boundaries rather than a solitary percentage, we shift our perspective from a simple headline to a more nuanced interpretation of evidence. It keeps us from overestimating the impact of a single study and helps us see the bigger picture. Now that we have established how these ranges define our uncertainty, we need to look at the specific mathematical architecture that dictates the width of these intervals, bringing us to the role of sample size and standard deviation in shaping our scientific conclusions. To understand why these intervals expand or contract, we have to pull back the curtain on the raw reality of medical data. The answer lies in the inherent noise of the world. First, consider the influence of sample size. If one person in a group of 10 has an extreme reaction, that single data point disproportionately drags the average in one direction. However, as we recruit more participants, these individual quirks begin to cancel each other out, leading to a tighter, more precise estimate. Beyond the number of people involved, we must account for population diversity. Humans are biologically complex, our genetics, lifestyles, and environments create a wide spectrum of responses to any given treatment. Conversely, a diverse group introduces more variability. While this makes the confidence interval wider, it is often more scientifically honest, as it reflects how a treatment might perform in the messy, real-world context of a diverse society. Whether it is a blood pressure monitor that is slightly off, a patient self-reporting their symptoms, or a lab test with a small margin of technical sensitivity, no measurement is perfectly absolute. These minute inaccuracies accumulate. It is important to recognize that this isn't necessarily a failure of the science, but rather a reflection of the precision limits of our instruments. When we see a wide confidence interval, it is essentially the data's way of saying that the signal is currently fighting against a lot of noise. By identifying which of these factors, small sample sizes, high biological diversity, or measurement limitations, is driving that uncertainty, we can better judge the strength of the evidence presented. Now that we have unpacked why data is naturally prone to shifting, we need to bridge the gap between these statistical ranges and the binary decisions clinicians must make every day, leading us into the practical interpretation of clinical significance. Moving from theory to practice, the width of a confidence interval acts as a direct indicator of the precision behind a study's claim. Imagine you are looking at a target. A narrow interval is like a marksman hitting the bullseye consistently, suggesting that the researchers have a very clear picture of the true effect. It tells us that while the researchers might have found an average result, the data is spread across a broad spectrum of possibilities, leaving us with a significant amount of ambiguity. When you encounter these wide ranges in medical literature, it is often a sign that we should approach the findings with cautious curiosity rather than treating them as an absolute certainty. Think of this as a boundary line between a meaningful effect and a statistical toss-up. If a confidence interval for a treatment effect crosses zero, it means the range includes the possibility that the treatment does nothing at all, or perhaps even harms the patient. It suggests that the observed benefit might simply be a byproduct of natural variation or random noise in the data collection process. Conversely, if the entire interval sits clearly on one side of that 0 or 1 threshold, we gain a much higher degree of confidence. It provides a shorthand for clinicians to determine if a result is likely a real, repeatable phenomenon rather than a fluke. However, a common trap is to equate statistical significance with clinical importance. We have to look at the boundaries of that interval to see the range of potential outcomes. If the lower end of the interval is so small that the difference is barely detectable in a real-world setting, the result might be mathematically sound but practically negligible. You are no longer just looking at a single number, but rather the entire landscape of probability that the researchers have mapped out. This distinction is vital, as it protects us from overestimating the impact of new interventions. This deeper level of scrutiny naturally brings us to the next phase of our investigation, where we look at how to weigh these statistical outcomes against the real-world complexities of patient care. Now that we have refined our ability to interpret statistical boundaries, we must bridge the gap between abstract numbers and the tangible decisions you make in a doctor's office. It helps both you and your healthcare team understand the potential breadth of a treatment's effect, rather than just relying on a single, misleading average. Imagine you are discussing a new blood pressure medication. If the confidence interval is narrow, say, a drop of 10 to 12 points, the evidence is quite precise, and you can feel confident that the drug will likely hit that target. However, if that same study presents a wider interval, such as a drop of 2 to 20 points, the picture changes significantly. By looking at these margins, you and your physician are essentially mapping out the best case and worst case scenarios for your specific physiology. This process is essential for weighing benefits against risks. If a treatment offers a modest benefit with a wide confidence interval that dips toward zero, it suggests that the therapy might not provide a meaningful improvement for many patients. In such cases, the burden of side effects might outweigh the uncertain gains. Studies indicate that when patients are presented with this range of possibilities, they often feel more empowered in their decision-making. It transforms a doctor's recommendation from a simple directive into a collaborative evaluation of risks. It strips away the hype often found in headlines and replaces it with a sober, evidence-based assessment of what you can actually expect. Ultimately, statistics serve as a compass, not a crystal ball. By recognizing that every clinical result exists within a spectrum of possibility, you become a more active partner in your own care. Yet, even with the most precise data in hand, there remains one final piece of the puzzle: understanding how these statistical insights translate into the long-term patterns of your personal health journey. To bridge this gap between individual data points and the broader horizon of medical science, we look toward the evolution of research itself. We are currently witnessing a shift toward more rigorous synthesis, where singular studies are no longer viewed in isolation. Through the lens of meta-analyses, researchers aggregate data from dozens, or even hundreds, of trials. This process effectively narrows our confidence intervals, giving us a much sharper picture of what truly works. By pooling these results, we can filter out the statistical noise that often leads to conflicting headlines, providing a more stable ground for the decisions you make with your healthcare provider. This modern standard of evidence-based medicine is essentially a collective effort to shrink uncertainty, ensuring that the guidance you receive is rooted in the most robust information available. As we wrap up this exploration, let us distill what we have covered. First, a confidence interval is not merely a range of numbers. It is a measure of precision that tells you how much weight to place on a specific finding. A wider interval suggests we should remain cautious and open-minded, while a narrower one offers greater confidence in the outcome. Second, these intervals remind us that medicine is rarely about absolute certainty, but rather about managing probabilities. Understanding this allows you to move past the binary trap of thinking a treatment either works or it doesn't, and instead view it as a tool with a specific, measurable impact. Finally, by looking for these intervals in the health information you consume, you gain a vital layer of protection against oversimplified claims. You are now better equipped to ask your doctor how likely a certain benefit is to apply to your unique circumstances, turning a confusing statistic into a conversation about your own well-being. This content is for informational purposes only. If you found this information helpful, consider subscribing to stay informed about how to better navigate the complex world of medical research and evidence-based health.